Hello, I'm Professor Kerrry Howell from the University of Plymouth, and today I'm going to talk to you about developing tools for biodiversity assessment and monitoring, and in particular about automated image analysis. So as I'm sure you know, the use of cameras has seen rapid growth over the last 10 years. We started with drop camera systems and towed camera systems and have now progressed onto things like ROVs, remotely operated vehicles, and also autonomous underwater vehicles, AUVs. And these new technologies are enabling us to produce quite vast data sets of both images and video of the seafloor. And these tools are increasingly used as a means to survey and monitor the epibenthic megafauna, particularly of the seafloor environment. But interpretation of these new data sets is extremely challenging. They really are quite vast, and identifying the animals from those images is difficult, particularly because taxonomic keys are not always useful with imagery. So traditional taxonomic keys have been developed to be used with museum preserved specimens. They're constructed from those preserved specimens and they're designed for use in that way. And they often rely on microscopic parts, for example, sponge spicules or holothurian ossicles or coral sclerites, as a means to confirm identification of a species. And these things are obviously not visible from imagery. And what it means is there is a lack of keys to identifying organisms using image-based data. And what this has resulted in is in individuals or indeed whole laboratories developing their own reference image libraries of morphotaxa. So what I mean by that is a reference library of organisms that can be consistently identified from the image data. And these organisms may be given a species name, a genus name, or sometimes even higher taxonomic level name, along with a denotation of morphospecies 1, 2, 3, or 4. So things, for example, like sponge species 1, sponge species 2. And these then enable people to analyze this imagery in some kind of detail, whilst being unable to put a confirmed species identification on things. The other problem is that when you analyze this imagery, it's extremely time consuming. So it on average will take about 1 day to analyze 1 hour of video, or somewhere between 20 and 30 images of the seafloor, depending on how complex they are. And this represents a major bottleneck in our acquisition of knowledge and data from these image and video data sets. It's also difficult for a human to sit at a machine for hours on end processing imagery. People get tired, they get bored, and they make mistakes. And their observers will have a bias, and different observers will have different biases. And this makes comparison of data sets extremely difficult. So computer vision and artificial intelligence offer a complementary way of analyzing imagery and video. Now, this is not a new idea. We use computer vision and AI in lots of areas of our lives. I think the most obvious are things like self-driving cars and facial recognition software, but we do use it increasingly in ecology and taxonomy, particularly in use of plankton identification. And there are now many open source tools available, things like TensorFlow, Keras, PyTorch, Deep Cognition, YOLO, and others. And they're becoming easier to use, although they still require a good knowledge of programming. So we in my lab have been looking into the use of these technologies in analysing imagery from the seafloor. And what I'm just going to show you is some very brief results from a research project we've undertaken previously. And this is actually published work, and you can see the reference there at the bottom of the screen. So we were looking at a data set from the UK's National Autonomous Underwater Vehicle, AutoSub 6000. And this data set consisted of about 60,000 images, which were taken from a single deployment of the AUV. And we annotated around 1,700 photographs from that data set. This work was actually done by Niels Piershaw, who's was a PhD student in my lab, so I just want to attribute that to him. So within that 1,700 images, Niels identified 40,000 individuals of 140 different morphotaxa. And then used this data set to train an off-the-shelf AI, basically. And so what we were interested in this study was understanding how much training data is needed to get a given level of performance out of the AI software. So how many training images do you need to feed it before it can reliably identify specific taxa? And the second question we were dealing with is how well does the AI do depending on how many taxa it has to choose from? So if you have a wide range of taxa, does it do better or worse than when it has fewer taxa to choose between? So essentially, we used our 1,700 annotated images and split that into training data sets of various sizes, and then tested the data sets as well using the same pre-annotated imagery. So we would split the data into 75% used for training and 25% used for testing the data set, and we would run many iterations of that, and each time testing how well the AI performed based on this human interpretation. And I'm just going to give you the headlines of what we found. So the main headline is that the more training images you give the AI in CV, the better it performs. That's not really unsurprising, but the important thing is that performance plateaus. So you never get perfect performance, 100% accuracy just doesn't happen. The highest we were able to achieve was somewhere in the region of 0.7, 0.8 performance. So that's 70 to 80% accuracy. The other thing that was important then is that the algorithm does better with fewer classes to choose between. So if you only give it 7 taxa to choose from, and you're only interested in identifying 7 taxa, it does much better than if you're asking it to identify 52 taxa. So you can see how performance changes in this plot here. The other thing is that performance is highly variable between morphotaxa. So the morphotaxa that humans do well on and find easy to identify, the AI also does well on. And the ones that humans struggle on, the AI also struggles on. So performance is at acceptable levels for some morphotaxa. And for other morphotaxa, it never does very well at all. So the main thing to take home is that these off-the-shelf, so they're freely available open source algorithms, have the potential to be useful for some taxa, and then could help speed up the process of annotating and identifying animals from imagery. But there are challenges. And one of the main challenges that still remains is the lack of standardization in the taxon identification between reference data sets that could be used to train CV algorithms. And what I mean by that is coming back to the point I made earlier, is that at the moment, each individual or even individual labs are designing their own taxon reference data sets. And what that means is that, for example, my anemone number 1 might be very different to someone else's anemone number 1, because we're not using a standard. And so what we need to do is develop a standard. And a group of us we've long recognized this problem, but back in December 2017, we managed to get a group of us together to discuss, to have a workshop to discuss the creation of an international standard for image reference library. And we published that work in 2019. This is the paper, which really sets out a framework for the development of a global standardized marine taxon reference image database to support image and video-based analysis. And we gathered together the reference libraries that have been created by multiple researchers and labs from around the North Atlantic. and we're in the process of merging those libraries together to create one standard library. And we also put together a schema of how we envisioned this database working. So the database would be visible through a web interface, and that web interface would allow end users to download things like a photo ID guide if they're going to see and need a book-like reference. It would enable a download, or rather an import, of a reference annotation scheme to image annotation software like Beagle, for example. And it would also feed imagery to things like the DeepSea ID app, which already exist. And then users will be able to download that video reference, image reference library as a database template, and then include new images of new taxa that they've seen in their own data set, and then submit those new taxa back to a central team who can quality control and then include those in the database if suitable to do so. So at the moment, as I mentioned, we are in the process of merging those reference libraries together to create the one standard for the North Atlantic. But in a project we have at the moment called the One Ocean Hub, which is working with developing nations to build capacity in particularly in deep sea science. We're working with South Africa, Namibia, Ghana, Fiji, the Solomon Islands, Seychelles, and also countries in the Caribbean. We are currently developing this web interface, and we hope to launch the database early next year. But we are also, as part of that project, building a South Atlantic and Indian Ocean image reference library under that project to support the work we're doing in the South Atlantic on that project. And so when we think about what we need to do to realize automated image analysis, to try and break this bottleneck of analyzing these vast data sets, there's quite a few things that need to happen before it's really viable. The first is the creation of a single global standardized marine taxon image reference database, which we are hopefully going to launch next year. The next then is a coordinated effort to improve the quality and coverage of images and videos in that database. So at the moment, lots of the images don't have a physical specimen to confirm identification. And what we want to see is people working globally to photograph an animal in situ, collect it, as you can see the ROV arms doing here, collect the animal, and then have that actual animal identified by a taxonomist as a physical specimen. It can also be barcoded, which will have relevance to the eDNA talk that I know you're going to hear. And over time, we will improve the quality and coverage of this image reference database. We also need to develop field keys, not just libraries of images. So a library of images is great, but actually we need real field keys, dichotomous keys like your traditional taxonomic key, but built specifically to work with visual imagery. And then we also need training materials for people coming into this field to have some kind of training in the identification of animals from imagery. We also need robust quality assurance standards in video and image analysis. So when you are wanting to pool data sets together, even if you have a single reference library, you still need to account for observer bias. And the only way to do that is to have cross training on each other's material and to ensure that those standards are compatible between studies. And then finally, all of that will give you decent data sets with which to train AI and CV. But actually, we know the off-the-shelf algorithms only do so well. So we actually still need to develop some better algorithms if we want to be able to apply this to more and more taxa. And I'll finish there. Thank you very much.